For over half a century, testing new drugs and vaccines on animals has been the bedrock of preclinical research. It is the established standard for assessing safety and efficacy before a compound reaches human trials. This chart shows the drug development pipeline, and it reveals a massive paradox. Millions of compounds enter preclinical animal testing at the top, but between 90 and 98% of drug candidates that pass those tests fail when they reach human clinical trials. Edinburgh Innovations specifically identifies this as a need for better human predictors. Currently, only about 2% of compounds progress through the entire drug discovery pipeline to a successful human outcome. Meeting the 3R principles, replacement, reduction, and refinement, requires moving beyond biological proxies and adopting human-relevant engineered systems. The consistent failure rate indicates an inherent biological mismatch. It is the result of using non-human species to predict human-specific immunologic. This translational gap is defined by the structural and functional differences between human and animal immune systems. Looking at cytokine expression, key human factors like IL-8, which is essential for neutrophil recruitment, are not naturally expressed in murine models. At the genetic level, murine MHC genes differ significantly from the highly polymorphic human leukocyte antigen, or HLA, system found in humans. Inbred laboratory mice also possess a limited T-cell receptor repertoire, failing to replicate the diverse TCRs found in human populations. Furthermore, mice raised in sterile conditions have naive immune systems, lacking the complex immune memory humans accumulate over a lifetime. Since these differences are hard-coded into the species' biology, simulating human-specific immunity requires a different architecture. The first layer of this architecture uses 3D human organoids, miniaturized models of organs like the heart, lungs, and brain, grown from human cells. These 3D models replicate adaptive immune features, such as germinal center formation, that cannot be captured in traditional 2D cultures. Researchers use microfluidic chips to introduce biomechanical strain. This lung-on-a-chip diagram shows epithelial and capillary cell layers, separated by a porous membrane for cellular interaction. Vacuum channels pull the chip's edges to simulate breathing, while an air-liquid interface mimics the lung's environment. These single-organ chips provide high local fidelity, but they remain isolated from the rest of the body's systems. Organoids and single chips are the components of the system. The next challenge is fluidic integration for systemic analysis. Systemic pharmacokinetics requires media to circulate through all organs without creating artificial concentration gradients. Standard sequential perfusion flows from organ to organ, leaving the final one with a vastly different drug concentration than the first. The Edinburgh innovation solves this. It uses a 3D printed device with a central pump and parallel perfusion, distributing media simultaneously to 5 distinct compartments. Interconnecting capillaries ensure drug distribution reaches the heart, lungs, kidney, liver, and brain, all completely simultaneously. This parallel flow mimics the human circulatory system. It allows researchers to use standard liquid chromatography to quantify drug uptake across the entire system. This clear 3D printed block enables the precise measurement of organ-to-organ interactions and systemic distribution in a truly human-relevant context. The final layer of the model uses artificial intelligence to analyze the massive multi-omics datasets generated by the chips. Deep learning tools like NetMHC-PAN predict peptide MHC binding affinities, allowing researchers to identify optimal vaccine antigens without animal immunization. While AlphaFold 3 can model complex biomolecular assemblies, it does not capture the dynamic conformational changes that occur during binding. Physics-based models are still required to predict how these structures shift as they dock. AI then integrates transcriptomics and imaging data to simulate the formation of immune memory in silico. The hardware provides the high-fidelity human data needed to train the algorithms, creating a synergistic hybrid pipeline. The multiscale hybrid model represents the convergence of human organoids, 3D printed multi-organ chips, and AI-driven predictive modeling. This integration results in a 50 to 70. This matrix compares traditional animal research against these AI-driven approaches. We are seeing a move away from in vivo challenge tests and toward in silico simulations of immune activation. For biomarker identification, researchers are shifting from observational animal pathology to AI-integrated multi-omics. This pipeline fulfills the 3R principles by replacing the biological proxy with an engineered human system. Global regulatory bodies are now formalizing this shift, moving non-animal methodologies from the laboratory into established law. The European Union's Directive 2010/63 sets the ultimate goal of entirely replacing animal experiments with non-animal methods. In the US, the FDA roadmap and the NIH's ORIVA office are both dedicated to validating human-relevant models to phase out animal testing. While challenges remain in fully replicating systemic immune responses, the path toward animal-free preclinical trials is now defined. The integration of microfluidics and machine learning allows preclinical research to finally align its models with the specific biology of the human patient.